This video will go over Unit 3 and maybe also, Unit 3. This video will go over Lecture 3 and maybe the part of Lecture 7 that we have covered. So let's get into that. This started with the idea that everything is everywhere. So by now you should be able to come up with some idea of why is everything everywhere and how does the environment select? So what does that mean? Everything is everywhere, the environment selects. Well, how does the environment select? It selects by having a specific temperature, having a specific amount of water, having a specific amount of oxygen, having specific molecules with carbon and nitrogen. All those things contribute to where can cells grow. So again, we think through this, and in nature, if cells have what they need, boom, they grow exponentially. If they don't, they try to survive in stationary phase and be dormant. They win if they can make endospores, because endospores have essentially unlimited potential for being dormant. So if you are a cell, if you are a bacillus, and your name is Clostridium perfringens, and you run out of nutrients, and you become an endospore, what is this process called? Pause the video. This is called sporulation. And if you decide, if you're an endospore and you decide the world is wonderful, I will become a cell again and I will make daughter cells and divide, that is called germination. And an endospore capable of germinating is called viable. So that's important terminology around endospores, and it's a good idea to think, what do endospores do? And what endospores do is nothing. They wait. But they are capable of waiting despite starvation, despite heat and cold, despite radiation, despite drying out, all those things. And the DNA inside of endospore is protected so well they can easily survive for years, if not millennia, if not millions of years. So they're kind of amazing in that way. So after we looked at endospores, we looked at temperature relationships. So I might ask you, where would you find a thermophile? Go ahead and pause the video. Where would you find a thermophile? Well, thermophiles tend to like best temperatures like you find in a hot water heater. So you'd find some in hot water heaters. You'd also find them in kitchens, places where foods can be hot and left out. You'd also find them outdoors in places where soil gets hot. So any place where a nutritious environment gets hot, like not red hot, but hot enough to burn a human, you might find thermophiles. And if it gets hotter, hyperthermophiles. Make sure you look at those things. But also, importantly, look at the shape of these curves. What is this axis telling us? It's telling us the growth rate. Thermophiles grow fastest at a certain temperature. They grow slower and slower and slower if it's colder and colder and colder. So study this. You don't have to memorize the limits, but it would be a good idea to memorize the temperature, optimum. So optimal numbers here. And then another question I might ask you is, where would you find an obligate anaerobe? Obligate anaerobe. Where would you find it? Go ahead and pause. Well, an obligate anaerobe, again, cannot grow if there is oxygen. It needs an oxygen-free environment. So where would we find that? You'd find that in a human colon. That's full of obligate anaerobes. You'd find that in mud at the bottom of a lake. If you've ever been out canoeing on a lake with a muddy bottom of the lake and you stuck your paddle down into the muck and bubbles came out, that's methane and hydrogen sulfide and hydrogen being released by obligate anaerobes in the mud. Anytime mud is flooded with water, that becomes a situation where bacteria can use up the oxygen faster than it can diffuse in to be replaced, so it's gone. So obligate anaerobes can thrive in flooded soil. So that's a thing. Make 100% sure you learn these words and what they mean. These are very important because these determine where we find microorganisms. Can they survive on our teeth? Can they survive on our skin? Can they survive in our guts? Can they do either? Yeah, make sure you know how to write facultative anaerobe if I describe a facultative anaerobe, or you describe it if I tell you facultative anaerobe. And then, again, practice interpreting these pictures. This, again, is thioglycolate agar, where we have put cells in and trapped them in place, and we've added thioglycolate, which reacts with oxygen. So oxygen diffuses in from the top and reacts with thioglycolate. It disappears. So very little oxygen ever makes it down to here. So at the bottom, there's an oxygen-free zone where bacteria can grow if they don't need oxygen. Up here, that's where the oxygen is. So bacteria that can grow if there's oxygen can grow there. Bacteria that grow well with oxygen will grow to a thicker density. So go back and review that. And then there are other nutrients, talked about a little bit. And then the growth media, there are some words for growth media, and you might review these words. So complex media, chemically defined, differential, and selective. These are different words to describe media we'd use in a laboratory to grow microorganisms. And what we've been using this whole term in lab are complex media. We've been using tryptic soy agar that is, it's made from soy protein that has been digested with enzymes to release anything that can be released if you chop up proteins. And the idea there is that lots of different bacteria can use that. So it is not selective. It lets as many things as possible grow, but it is complex. We don't have a list of all the molecules that are in it. If we had a list of everything in it, that would be chemically defined, and that would be like this. This is chemically defined medium for growing some E. coli strains, where we know exactly what molecules we've put in. And in this case, it's super restricted. There's only one source of carbon. So every lipid, every amino acid, every nucleotide, every carbohydrate that gets made in this cell is made from carbon that originally came in the form of glucose. Every amino acid and nucleotide require nitrogen from this ammonia. That's just really something that cells can do that. And then another word we found was fastidious. Fastidious. Fastidious refers to a cell that needs lots of nutrients. So it wouldn't look like this. Remember, something like E. coli can convert glucose into so many different molecules. It can make every vitamin it needs. It can make every enzyme cofactor. It can make everything. A fastidious organism cannot. A fastidious organism is used to living in a place where it finds those things. It finds all the amino acids. It finds all the vitamins. It finds lots of other different molecules, and the pH is normally buffered very nicely because this lives in human tissues, and everything is very well controlled in human tissues. You know what else needs a recipe like this? Human cells. If you try to grow a human cell in a laboratory, you need to give it something that looks like this, a long list of molecules, because they are incredibly fastidious organisms. Human cells are. And it's fine, because the whole body is designed to give them all exactly the environment they need. Whereas there's nothing designed to give E. coli this exactly. So something like E. coli is ready to grow in different environments. So then we go back, what was selective? What was differential? Well, selective media restricts growth. It only lets certain things grow. So an example of that is McConkey agar, which has crystal islet that kills gram-positive cells. So it only lets gram-negatives grow. Differential media tells you something about what's growing in them. So blood agar is differential. Cells that can do hemolysis, their colonies look different. And McConkey agar is differential. It has a pH indicator in it. So that was lecture 3, and it has turned out to be a pretty long one, so let's end it here.